Hello, everyone. We have a very special guest and a very special angle to today's meeting at the Biocreation Station. We will see unique perspective of scientists who will choose the works from the Biocreation Station that they caught their eye and they provoke them to think. So we will hear from Kenza, Eloise, and Stephanie. And later we will have a presentation of works by two bioartist designers, Heidi and Carla. So without further delay, I give the floor to Kenza. And do you want me to share the screen, or will you share a screen and like do all the things by
If you could share the screen,
okay.
I had the impression that there would be a tour and then I could comment kind of like live. I didn't really prepare something for this, so but
Okay,
we can
no worries.
go ahead. Maybe if we yes.
Maybe you can also like introduce like a little bit
Yes.
yourself.
In the meantime, I will introduce myself. So I'm Kenza, I'm a PhD student and I work on microfluidics, so manipulation of fluids on the microscale. The reason why I do this is I basically design devices to automate parts of the workflow that scientists do when modifying mammalian cells. So in this process, when you're gene editing mammalian cells, whether it is to develop new cellular therapies or whatever, there are a lot of steps involved to kind of create a cell line that's clean in a sense to say it. So all cells are exactly the same and exactly the same modified. And in this process, there's like lots of machinery that you need to use, and this can kind of like harm your cells as well. So in a way, I'm using microfluidics as a more holistic approach, I guess, to to say it in an artist way, to manipulating cells on like a single cell level. So I guess that was my intro. And maybe the way we should do it, since I didn't prepare anything, Carolina, how about you just pick a work and I'll give my perspective on it.
Okay, maybe it will be interesting to comment on Carla. She's going to speak later on about her work, but I think it can be interesting for you, or maybe maybe I have an idea that's like a very live situation. Maybe Carla will speak about a little bit about your this work, and then you will have your insight, because it seems like by printing and blips and blobs, it's somehow like I related it somehow to microfluidics. It doesn't have to do, I think, a lot with cells, but maybe it does too. I don't know. But Carla will
talk about
it.
Yes, sounds good. I can talk briefly about it, and if later on people have questions, I can show more pictures and videos that didn't fit in the gallery. So briefly, blips and blobs is part of the research that Nantini, another researcher, and I did at Brooklyn Research, and we had a prompt. So the prompt, it was new forms of data representation. And it was very interesting because both Nantini and I had kind of like opposite ideas of what our starting point should be. I was very interested at the very beginning on the larger scale, on man-made systems such as electromagnetic signals. And Nantini was super interested on the interference of living systems, in particular, how we could interact with slime molds. So after like very, very long conversations, we decided that for us the connection was how could we interact as a man-made system and interfere with the living system. And then can you please show the picture of the whole system, like the one that has 5 objects? Yes, this one. So in order to achieve our goal on creating a visualization using living systems, we somehow needed to create a pipeline to make that happen. So we ended up customizing different, let's say, technologies that already exist to fit our purpose. So we had an antenna to capture satellite signals live. So we did different recordings of NOAA weather satellites. And then the recording is an audio file. So basically it has features as any other audio file. And we created an algorithm that would process every individual audio file to be able to be represented in a physical space. And the physical space that we chose, it was a petri dish. So the algorithm converted an audio file into coordinates inside the petri dish. And we wanted to have accurate coordinates inside the petri dish, and that's why we ended up hacking a 3D printer that would allow us to have that precision. So the 3D printer, and there's another picture with more detail, it's connected to a pump with a linear actuator that would help us have like super precise droplets of oat space that we would position inside the petri dish to let the slime mold grow towards the dots. So basically we created a system so the slime mold would grow towards the coordinates that we have previously decided. And at the very end, we wanted to make sure that when we did the exhibition, people could observe the final display. So there were 6 petri dishes that had the different visualization. And maybe if you could please move the pictures of the slider, I can say, so this is one of the final results. So there were 6 like these. There were 3 different satellites at different times and at different days. So we wanted to keep the essence of the kind of like our geographic position. We were based in New York at the time. So it was very important to see that this representation only makes sense at that geographic position. The next picture, please. Yes, so what I explained is basically this. I decided to add the whole pipeline to make sure that if anyone was interested, or if anyone has any question about any of the processes that took place, this is basically the whole system that we put together to go from the satellite signal to the petri dish and the slime mold pattern. Next picture, please. Yeah, and this is the kind of like the promotional flyer that we had when we opened the exhibition. And this is what Blips and Blobs means. It's visualizing the largest scale of invisible satellite signals into the tiny scale of the slime mold. And last, there's a picture of the 3D printer and the pump. Yes. So this is, I would say, the core technology that allowed us to go from the digital wall to the physical wall. And even though it's not perfect, because I'm a creative technologist and designer, and Nan is an artist. We kind of like figure it out on the way. We believe that it has a lot of potential with other signals that can be represented as well in the physical realm with living systems. So I would say overall, this is all.
Nice. All right. I was thinking I could comment as in like opportunities for like more or different, I guess, technological improvements. I don't know, it's not improvements, that's the wrong word, but that you could try out. But I'm not sure if that's that useful. Maybe I'll give like the perspective of how I see this. Maybe like if I'm a scientist and I would go to a gallery, this would immediately attract me. You know, I see a bunch of syringe pumps and yeah, I think it would immediately attract me. And but I would have a hard time like trying to understand the main question of your work, like why was it important for you to take data, and why specifically satellite data, I guess, and transfer that into like a slime mold structure.
Yeah, maybe that one
I think the satellite. Go ahead. I mean, sorry.
Satellite data for us represented something that it was beyond kind of like the boundaries of Earth. So it was something that it was accessible, but at the same time, it was such unreachable for us. And we decided to go with the NOAA satellites because we knew it could work. So I guess that in the future, it would be amazing to try with other signals and see how the algorithm reacts to those sound signals.
No, that's amazing. Like when you explain it in that way, I totally get it. So I guess it's like on a different perspective. Yeah, that's super cool. Like translating something that's kind of some form of data that we are not directly connected with on an everyday basis. I mean, I guess we are through internet or whatever, but it's not something we can grab or feel or and then putting it into something like biological. That's awesome. Yeah, that's really cool. I love this. I would be immediately attracted to this if I would see it in a gallery. Is there so you shared like a diagram of the system, and I thought I saw that you can also like transfer pictures or something to it. Could you share that again, Carolina, the overview of this system?
I left a link on the chat because there's a lot more documentation on my personal website, and maybe some of what you're missing is there.
Nice. Okay. And I think it would be real. Is this Python code also open source?
So it's not available yet, but our idea, and I know it's we kind of like finished the project 1 year ago. We are still figuring out how to make the whole system available in a way that it's easy to make it work when you try it. So we are working on it, but it's not available yet.
Nice. That would be awesome. I could see this as something people could also change the code in, right, and push it, like if you would work with GitHub or something and then change the code and then there's an update of the system and people can print new stuff and such. It would be so nice.
Amazing.
Okay, thank you so much for sharing your work and your insights, Kenza. And maybe now we will invite Stephanie to speak about the works that she chose. Hi, Stephanie. Great that you joined us.
Carolina, can I have a question? Hi, Carolina, very quickly. Heidi has a question about Carla's work.
Oh, okay. Sorry, I didn't see that. Heidi.
Thank you, Carolina. Thank you, Carla and Kenza, for talking and showing your work. I wanted to know if it was difficult to, I know slime mold grows very well, but did you get a problem like in contamination and stuff like that? How do you set up because I just took a picture and it looks really good, like the color and how it grew and went to the different like hubs of activity in the same to capture all of that. Did you get contamination on your?
Yes, a lot. And like if you were to go to my website, the final picture that we got is molded. So we decided to add some resin to kind of like freeze the state of the slime mold, and it was already molded. And if anyone wants to explore these and using oats paste, the main problem is humidity. So we had to grind the oats into a very fine form so it would flow inside the pump. And that was the main contamination, the main problem that we have with contamination, because of course everything we tried to use, it was sterile, but when we added the oats that it was grinded elsewhere, there was always something that would mess up some of the tries. And then water was our worst enemy, because if it was too dry, then it wouldn't flow and the droplets would be kind of like grimy thingies. And then if it was smooth and perfect, then it would mold before the slime mold would grow. So I think that we needed a more sterile environment to make this work. So one of our ideas was to make this system work live. We didn't get to that point, but whenever the satellite was passing on top of New York, kind of like record the signal, process it, and then print on the Petri dish.
And another one would be, how long did it take for the slime mold to make like this path? How long did you leave it for how long was the signal being recorded with the fungi, something like that?
So in 24 hours, there was quite a decent network, and it was when it wasn't molded yet. I would say that when it was super interesting visually, it was in around 48 hours, but that's when our intruders would be also in the Petri dish. So it was like finding a little bit of balance between more complex network and less contaminated.
Great. Any more questions before we invite Stephanie? No. Okay, thank you, Carla, so much, and Kenza and Heidi for very thoughtful questions. So one more time, hello, Stephanie. Great that you're joining us. Please say a few words about yourself and your background, and what would you like to talk about from the biocreation station? What works that caught your eye?
Hi, can you hear me? Oh, okay. So I'm Stephanie Okeo. I'm a microbiologist, but also a science communicator and engager. I'm based in Kenya. That's, I think, in brief what I can say about myself. And I think the project that caught my eye was the bleeps and blobs. First of all, I like the name. It's catchy. But the fact that I looked at it, I don't know, I'll give my perception as somebody who's trying to communicate science and make people interested in the project. So it's a unique project in terms of it's bringing in different parts of science, technology, and innovation within one project, with the microbiology aspect, the satellite, and the radio and frequencies and all that. I think, but I was wondering, why just slime mold? Have you tried different organisms? And even it can be a good way to even teach different types of organisms and characteristics, because they have different colors, different shapes. But I think also the other problem is contamination, which is a headache. So how do you get pure cultures within that space of still transmitting data? So I felt that really interested me. And it's a good way to teach people about different fields of science and technology. And with Kansas, I did get an opportunity to really see it well. But I saw this, the bra and the panty thing, and I felt like it was really interesting with, I think it was written somewhere, pregnancy. I didn't get a chance to to look at it. Those two will have been very interesting because anyone who sees, especially male or female, with that kind of gadget will be interested to know what the hell is this. And with the blips and blobs, it will be interesting to see, okay, what is making this? Somebody who doesn't know science, what's this thing growing? And it's something invincible that's transmitting data to create something that we can now see. So that's my take. Don't know if it makes sense. Everything we do here sometimes does not make sense. So yeah, that's my take.
Oh, sorry, I'm muted. Thank you very much, Stephanie, for your insight. Feel free to jump in at any time and like share more of your thoughts. And now I would like to introduce Heloise. Probably you remember her.
Sorry, I have a question.
Sure.
A question. With the cancer, the outfit, I didn't understand what it is because we were scrolling through the page. So I just wanted to know, like in brief, what exactly that.
I think Stephanie is interested in the consumer pregnancy reports. I think that's the project you're talking about.
I think so, because I joined when now the page, we were scrolling through the page.
I think it's that one.
Oh, this one, okay. That's Annie Liu's work. Oh, let's, I will share my screen then. Wait a second, guys. So this is Annie's Liu work. That what you meant, Stephanie?
No, that's not the one. There's one that has a garment looking like a bra and a panty. I don't know. I think I saw my own things maybe.
That's this one. That's this one. It's like later on. It's here.
Oh, okay. Yeah. Yeah. So that's what caught my eye. Okay.
So what did you think about it? Did you dive into this work or?
I found it interesting. It's like competing with the lingerie word, but like there is some tech in it. And I just thought about it and was like, this must be really uncomfortable to wear. So I was just with the thoughts of, okay, what is this? No biological input, but I think it was just interesting. And these small things remind me like something of a cyst. I don't know. It's just so weird. So I was interested to know exactly just what it is.
Unfortunately, we don't have an artist with us, but this is like, as she states, ritual devices and consumable products related to the pregnancy. And I think it's just like a series of works that try to address like milk pumping and all like the body changes and hormone changes in the body during the pregnancy and after. So probably some of these works are speculative. I'm not sure what's inside this vial, but it says mood swings, so we can imagine that like those pills are containing
hormones, something of the sort.
Yeah, hormones, exactly. And here we have oxytocin inhaler. So like to be more bonded with the baby, and for it addresses somewhat, I don't know, like this baby blues, I think it's called, like when you have a baby but can't relate to it, and like it feels like a very distant. Like more of the.
Have you shared the link?
Yeah, sure.
I just share it.
I have a question as someone who doesn't know that much of art and design, I guess. But this is like, to me, like really appealing. Like it looks like product. Is this like a specific type of like art stream, or I'm not sure how to call this, but like everything is like designed as a product and as if it's like an experience, and I could just go to the store and like buy this product for getting stretch marks. Not that I would ever do that, and that's like the fun part here. If someone can educate me on that, that would be awesome.
I think they might be designed like that in order for us to put it in something that we can relate to. Like you were saying, like if they look like cosmetic products, but when you go into the description, it goes into something that you have to like make a reflection on. Would you like to use this to produce stretch marks or things that are not visible, like in the whole pregnancy process, but then they're making it visible there. So swelling, these are the things that are going to happen during the pregnancy, and maybe they're overlooked. And as Carolina was saying at the beginning, like the pumping, and you become like this, in a way, machine of feeding and maintaining a human being, and there's maybe power into it. And then you get all these things, and probably in how, maybe now it's changing, but how it has been currently, how it's being treated. I'm not a mom, so if there's a mom in the room, that would be very much talking from experience. But I think that they're just like, oh, you're just having mood swings, it's fine. Like just in a way, just like deal with it. It's just your hormones, but it's so strong. Like my sister had her daughter a year ago. It's not something that you actually can control. It's like your body changing with all these things. It's not like you can choose, okay, I'm just gonna get the stretch marks and I'm good with the mood swings, it's fine. So I think like just having this conversation, it's what the work it's for. It's just putting it on the table. Okay, let's talk about this. This is not something that just passes by. But I'm not a mom, and I'm a designer, so it just.
Yeah, can I comment?
Right, yeah.
Yeah, by reading the stuff that is written on I forgot how to say it in English, but the bottles of the products, these are like all uncomfortable stuff that people that are going to have babies go through. Like oxytocin is a hormone that makes contractions on your body so you can like push the baby out. And there are like syringes with hormones. So I guess the point was that no one by free will would use these products. You know, there's many products like to erase stretch marks, to beautify a woman, but there no one is talking about these processes as beautiful or as desirable. To have like mammary leakage, I can't read it, like to your boobs be dropping milk, you know, no one wants that. And why does no one wants that? I guess that's the point.
I mean, like
It also, can I add in something? I think it reminds me also of, it reminds me of how nowadays people use their placenta, put it in different, and make it to different products like face cream creams, peels. I think it's just the same way how people nowadays use their placenta to and modify it to different products. So that reminded me of that also.
That's very interesting association. Yeah. Okay, let's. It's also like very aesthetic, like this beauty aesthetic and this like designer touch that Heidi mentioned. I think it's like it's in a nice contradiction in with the content, because this is not right desirable thing to have mood swings. It's rather something that you want to avoid. But yeah, I'm thank you, Stephanie, for choosing this work. If you have any other work that you would like us to show and discuss and comment, please do. You can do it now or like even like slightly later on. So so please let me know. No, okay, you're not speaking right now. I thought maybe you're muted. So I will move maybe to the whole panorama of Biocreation Station and invite Alois to share her scientific / philosophical insight. So great, thank you, Stephanie, for being with us. And if you will want to pop up with any new work later on, please do. And now let's hear Alois. Please tell us a little bit about your background in science, and I admire your love for philosophy. That was really great, like your very passionate reaction during the Andy talk was amazing. And yeah, and it actually brought like to the BioSummit this level philosophical dimension that it wasn't so present for me in the last BioSummit and 2 years ago. So I'm very happy. Please.
Yeah, I'm so happy with that comment. It's amazing to me to be able to bring this perspective for you all. Actually, my name is Eloisa Osbal, but I accept any variations of my name because I know it's tricky to say it. I'm a biotech undergrad student. Actually, I'm from Brasilia, Brazil, the capital of Brazil. And since my high school years, I've been reading philosophy much for an influence of my mom. She works with education and technology, so the books were always on the, I forgot how to say it, the for me to reach out and explore them. So I have I've been reading Deleuze and Guattari. I don't know if you guys are familiar with them for so long. I don't know, maybe since 2016 when I was 16. And I've always tried to merge those concepts and those ideas with my scientific interests. So during high school, I always enjoyed chemistry, physics, biology, and then I hopped on biotech undergraduation, and I never stopped reading philosophy and doing courses, but it was always like a barrier to me to be able to bring those discussions between my biotech colleagues, because I think that it is so essential for us to be talking about philosophy and ontologies and cosmologies, because what we do as a scientists and on the science, it has like a really strong Descartes base. So to see the world like as an objective reality, and we are the beings of reason, and we have a hierarchical view of the world, and that is the base from all the work that we do. And I'm not saying that science is bad by nature or something like that. It has a bad essence, because look at the BioSummit. I mean, it's so many amazing people, such an incredible space. But I think for us to be able to push these changes that we aim to, we need to look at our own ontologies of the world. So I chose two artworks to speak today. It's Hybrid Matter by Daria Warner and Bacterial Dyes, I'm sorry, by Cinzia Ferrari. But actually first I'd like to tell you more about this, how to maybe tensionate our ontology of the world. So I would like to start with an exercise. I would like to, if you can, for you to close your eyes and imagine that you are at the forest. Any forest, okay? Any forest or your regional forest, or I don't know, a forest that you created in your mind. And I'd like to think about, what do you see? What objects do you interact with? And to please name them in the chat for me after a few seconds of reflection. Feel free to name everything, anything that you want. Cool. Grass, bark, moss, chanterelle, mushrooms, water, pebbles, trees and rocks, snail, the scent in the air. Cool, the scent in the air is cool. Okay, perfect. So let's stick with the tree word. You see, the tree nomination was essential to be able to move around space, to know maybe that it can be a food source, also to tear it down and use it as a building material. The thing is, we all, sorry, I have like a little script here. Yeah, I see everyone named many stuff, that's great, but let's focus on the word tree. And I'd like you to think with me. Someone or some group went to a forest for the first time ever and looked at this crazy diversity and started naming its stuff. For example, they looked at some hella big brown thing with small green things around, maybe some colorful dots, and named it tree. You see, the tree nomination, to name a tree, was essential for us to be able to move around space, to know exactly maybe that it can be a food source, and or you can use it to build your own house. The thing is, we are that this small slice of people between all the diversity from the world. We recognize tree as an isolated entity up to this day. We nominated it isolatedly from the forest as one of its composing beings. But it wasn't nominated as it emerged on this world. We did it, right? We did it mainly for purposes of navigation between this chaotic world, because no one can survive. Like, I know a little tale by Jorge Luis Borges that it's about every time he arrived at his home, he didn't recognize his dog, because every second his perception about his dog was changing, because his dog was changing. Things change over time. That's the flow of nature of time, right? Okay. So we may say the tree happens. This is important. The tree happens to us in an isolated way. We created a conceptual representation of what a tree was, and many times whacked by encountering diversity and trying to fit all these trees' diversity in the concept, in the model. We have it in our minds, right? And as the list says, an event is a mixture of bodies that we can recognize. The notion of event that is connected with the happening, the happens stuff, by Deleuze means a relationship of knowledge with the world that it's opposed to the representationalist perspective, where the entities of the world only acquire its existence in action. That means when I go to the forest and I look at a tree and then I name it, like it is happening to me right now, right? It is not a matter of investigating a given essence or being, but of understanding how certain things happens to us. Subject and object compose each other. So we can, by this conversation, we can think about a case that happened here in Brazil between 1967 and 1970, that two American researchers came to Brazil and went to the Yanomami people, and then they collected blood from them to do research, and then got back to the United States and kept those samples frozen over time. So what happened was that blood was frozen on those freezers, but the Yanomami people have a culture that when people die, they have to destroy everything that belonged to the people, or that even made some reference to the person. So they treasure their bones and they burn everything. So it was a real issue for them to know that the blood samples of the people that were dying were frozen, because that was a piece of their body that was still here in this material world. So you see, we can apply this notion of the event to understand that it is not like the scientific purposes that will be superior to a certain Yanomami view of their need to destroy all the evidence of their people that died, you know. So that's why this is really important for us, that we do science. Yeah, so wrapping it up, we see diversity. If there would be an essence to this world, it would be diversity. Everything is diverse. We name it, we recognize it, we create a concept about it, and we try to use it every time we need to think about things. Let's think this in terms of what Drew Andy was telling us Friday about how to build a positive future. In my opinion, we need to change our own ontologies and understand that things aren't what they are. They aren't complete, and we just go there and use them passively. You know, we are not just like scientists that use machines. You know, as Manu Prakash said yesterday as well, a pen doesn't tell you what to write. And this is exactly, this can wrap it up, this notion of event and the happening in the loose. A pen doesn't tell you what to write. Sometimes you can be using a pen to write on a piece of paper, but you can use it as well to kill someone. I don't know. So subject and object are always composing mutually, right? So going back to the works of art, bacterial dye is not only isolated a soil bacteria to produce dyes. It proposed a new encounter between bacteria bodies and fabric bodies, and they developed together something that we couldn't expect from the beginning. This is important because the things aren't always contemplated by its concept. You know, we can't, many times we can't expect what is happening, if we're going to have a positive result or a negative result. So it's a mixture of bodies, and that's why this is so beautiful. So here, Gentylobacterium lividum went through an event that actualized its potencies about what it is itself and what can it do on this world. So we can talk about Gentylobacterium lividum as the concept we had about it and try to fit this new ability to it to produce fabric, to produce colorful fabrics. You see, and we produced a new sense to that bacteria that wasn't given before. And also look at how companies dye fabric up to this day. This didn't happen from day to night. It was a process. And this is also a major problem when we take things as like transcendental, you know, like this is the transcendental way to make colorful fabrics, and this is what you have to work with, and everything like all the other new ways to dye fabric, maybe they're always like peripheral, and this is a really problem because it's not about it. This is a new process of creating a colorful fabric that couldn't be predicted by this theological way of seeing the history, and that we don't know if this is going to work out, if we're actually going to use this as the way to dye fabrics in the future. But the important is that this is an experimentation that involves so many views, so many ways of living, and that it's actualizing, I guess, this way of living of the Gentinobacterium living in this, wow, crazy, so awesome. Yeah. So now moving to the art piece Hybrid Matter, something similar is happening there. Like,
can you please sorry, the name of the second work?
Sure, it's Hybrid Matter.
Hybrid Matter. Okay, I have to find it. Oh, yes.
Yeah, that pink stuff. Yeah. If we look at that art piece, something similar is happening here. As it is written in the description, slow encounters with mycelium mind, the encounters. Anyone that knows Spinoza may understand why I'm so fixed on this concept of bodies. The artist proposed herself to encounter mycelium and residues. Let me see exactly. Factory clay, coffee grounds from a local coffee shop, and cardboard board from delivery boxes. And it actualized the notion of 3D printing and also put together the mycelium. So she approached them in a radical experimentation, and something happened in between this gorgeous and hybrid artwork. Yeah, and I love mycelium. I've worked with it for a few occasions, and I think this is really awesome. The pink color with the gray stuff, crazy. So wrapping it up, this was my point. We shouldn't see creativity and art processes as the sum of things we already name on this world, but a rather radical exercise of seeing the chaos in things and dare to mix them, always producing the amazing new that isn't limited by the concepts we have in mind. So we think creativity always starts with like integrating a tree and paint and biomaterials. So we unite these things to produce art. You know, we have these sommatory ideas in our mind that are essentially in our own mechanisms to navigate on this world. We shouldn't be extending these to all this diversity and this this crazy world, you know. So yeah, I guess that was it. Deleuze is always talking about this concept of this conception of the world based on the idea of multiplicity, because everything is multiplice, because it modifies its being, its way of being, depending on the array in which it is integrated, it is put, it is interacted. Yeah, so I guess that was it. If you have any questions, any comments. I don't know actually if I was able to express as concisely as I was expecting, but because I'd never said these things in English, so I'd love to hear from you. Thank you.
Thank you so much. I loved it personally, but and I think like you made this concept of like this radical exercise of seeing a chaos, like and making like ordering in a different order than had been set, and this very like vitalistic Spinozian touch in your speech was wonderful. And but let's open up a conversation to our guests. I mean, to you guys. There was like nice comment from Georg. He said, like regarding positive futures, but I think it's like loosely relates to the speech, but it is like there are many bad dreams about biotechnology, but someone also needs to have the good ones. We artists need to have the good dreams. Quote Joe Davis.
I think it's important that the artists have the bad dreams too. Because me as a scientist, I feel like I only like to think about the good things. Of like for me in my field of synthetic biology, so and I think many of us don't like to think about what bad our things can do, as in like ethics. So I really like the provocative and weird and art that points at the bad things that can potentially happen. Yeah, that's my perspective.
Thank you. Stephanie, do you want to like add something maybe from your unique perspective on the vitalistic Eloise and dream quote?
Oh, yeah, I guess that it's exactly that. Like, there in Netflix, there's this new documentary that's all about saying that social media is really bad and it's destroying our future. So there's that like that guy from Facebook that helped developing the technology, telling that then he had his small children around waiting for to play with him, and he was trapped in his cell phone by that so evil technology he created. You know, and I guess that brings so much anxiety for the young people who see it, because they tend to think and like expanding the social media and taking social media as a technology, right, and expanding that to science and all of that, because they tend to think like that everything is given, that everything is ready on this world, and it only takes us like we are a piece of this crazy system that are oppressing. Of course, there is so many oppression, there are so terrible things going on, but we also still have that potence, I don't know, yeah, potency to look at things and to think, okay, this is the major way of using these things, like the bad dreams about biotechnology, would that be? But how can I transform it using my contacts, connecting with other people, integrating maybe notions from the arts, from other scientists, other science fields, and to make it always a creative process of combining bodies and experimenting and see what we have as an output. Always looking at the force side of things, going more to Nietzsche point of view, to always be like analyzing what forces are in the game, so we can actually move around and be more sensitive to the things that are going on. Because if we analyze the case of the Yanomami people, many people were involved on that process of removing the samples and then going back to the US and working on these samples, and their genome was actually used on the Human Genome Project, so it is online forever. And those people are still bothering them, you know, because they weren't able to move from this plane to go to the other, to the dead people kingdom. I don't know, but this is I think with this perspective of creativity and knowing that the things aren't given to us, we can be more sensitive to not join these kind of things. Because at that time, the people were like, oh no, this is science, we need to advocate for science, and science as the transcendental unit that is essentially good and all of that. So we need to be sensitive about these things and keep on having the good dreams.
Thank you. Thank you a lot, and I look forward for like unfolding like this bio-philosophical path in the future bio summits. That would be great to have some kind of like a philosopher's command here. And now we will hear from Heidi and her biomaterial dreams of the future. So let's.
I will share the screen.
Awesome.
Okay. So this is actually, I'm an industrial designer by training. I changed the title to experimental designer because it sits better with me. And it's been 2 years now since I put together this group of working on material. I don't know what do you see? Just the screen? Do you see my screen?
Yes, we see all of the screen, like this here, like a desktop.
Oh, is that better?
No, I still see all of your desktop.
Okay, sorry. You should be looking only at this. Now, is that better?
Yes.
Great. So this is the work that I've put up at the gallery. So we're an interdisciplinary group. It's made up of four biologists that are specialized in mycology. And then there's designers from the fields of textile design, industrial design, graphic design, fashion design. And we all come together in this group to just start taking material as an excuse to start going deeper into this topic. And I realized through our work that I'm not particularly interested in the material itself, but just the material, what the discussions that come about building this material. So this work that I'm showing, it's called Chromatic Expressions, and what we wanted to explore was how to express color through these fungal biomaterials. And at first, what happens is the first idea that came to mind on us was to explore it through adding color, which is how designers might be the first way to look at it, like dyeing fabrics. So we started on that. So I'm just gonna show you guys a little bit of what we did and how it changed the direction of we were going. And we work like this. We're based in we started as a group outside of academia, and then we validated after a year of that. And this is FADU in Argentina. So there's two buildings where the design building and the science building are next to each other. They're just like the same building, but differently, super different inside. So sometimes we meet at the Faculty of Design, and sometimes we meet in the lab when we need stuff. And we started doing these workshops about biofabrication for the first time in 2018. And there wasn't a lot of things happening around in Latin America and in Argentina around this topic. And we opened it up for everyone, because I think this is so interesting about this, what is happening with biomaterials is very much bottom up. So you could do it even if you don't have the background, because it's easy to learn it while you're doing it. And in this first, we explored the variable of morphology through computational design and digital fabrication. And after that, the question that came up was, what about color? What happens to this variable? And also the thing about calling this type of creating materials, like these new materials. It seems that they knew, but this is really interesting, an information report of the CIA. And you would be like, why? From 1955. And what they're saying here is that myco wood, it already existed. There was a CIA report on it on 1955. And this is really interesting because it's like, oh, we're building these new materials and stuff. But then I went into like more research and found out that in Germany with this paper, and around 1930, this guy already was working with myco wood, and about 120 million myco wood pencils were produced on the GDR from 1958 to 1961. There was already a fabrication development made of those years. And my question there is like, what happened? What is the lobby? What is there happening with the companies that didn't make it, and we're just seeing it like 10 years from now, like 10 years back from now? This is how it started to grow. But this has been happening for more than 30, 60, I don't know how many years, I'm really bad at math. But the thing is, are these things really new? And I think going back to the reflections of indigenous and other type of explorations around materials that have been a long time here, and now we're just like rediscovering them. And what we're doing with the chromatic expressions, like I said, we started looking into dyes, because this is what the first input from the textile designers when, okay, we have to dye this, we have to dye it with natural dyes, so we're using from cochinilla. And since the fungi surface is hydro repellent, I don't remember the name, hydrophobic, we use oil-based and water-based dyes for this. And the fungi that we're using is Ganoderma lucidum, and we just started doing some tests with it fresh and with it already dried to see how the color will incorporate on the material. And what was interesting, again, this type of things that you come to other research through the experimentation in design. So what happened in this is a different variation of the same bread, but fungi, this type of fungi is really good for decomposing dyes, and they're using it in a research in the lab to clean up water sewage from the fashion industry, which is one of the most contaminated industries out there. So it's interesting. So this organism is not only like we're looking at to produce color, but then we're seeing that it degrades color, which is an interesting take. So what we did, okay, we can dye it, but wouldn't it be interesting to have the organism express the color itself? So we look into 3 different species that we could that we try to see if it would express the color in different substrate. And that's another like input thing that happened. Okay, this is not like in design, we just wanted to make like a color palette. Okay, and then if the fungus is yellow, then we're gonna have a yellow thing. But it's not gonna express itself in all the environments. Also, we didn't get anything in the solid substrate. We did get something in in the liquid substrate. And also what was interesting is that what we understand about like fungi is not the fungi itself, but the mycelium. The basidium or the fungi, if it has color, that doesn't mean that the mycelium will be color as well. So there's research to be made also, which are the fungi that have colored mycelium. Then we went into this one, Cerocomelus, which didn't express in any other substrates, but just in the agar-agar. So we didn't use it. At the first, we wanted to use everything. And then we came into Pycnoporus sanguineus, which it did express it. And what was fascinating about this is that in the samples, it happened by mistake. We realized that if there was a change in oxygenation, if it didn't have any, like a lot of oxygen, it wouldn't express the color. So that became a variable that we could also use in order to produce new things. And this is one of the molds from one of the people in the group. To the molds, we put like little holes for it to breathe in order to grow. And when we took it out, because then we let it colonize properly, in like wide open out of the mold, you can see here that the holes that were made for it to breathe, it would produce the color, but in the rest it wouldn't. So now we can use it in a way like a printing organism, where we can make like a stencil, and we can decide where to have the color. And this is actually what happened when we left it open, and it finished colonizing the whole thing. And again, science counts, because with designers, we're like, okay, now we have 3 different fungi that had 3 different colors. Let's mix them, and then let's get a bunch of new colors. And then the scientists were like, the biologists were like, okay, no, they don't mix just because you want like have to mix colors. And he gave us this paper like they are territorial and they won't get from one place to another, and was like, okay, we can't work with that variable, it's fine. And so what we wanted to have at the end was a color palette in which we can explore this variability, because also at first, as the designers, we were looking like for this standard color that we can replicate over time. So it was a really learning of these new paradigms of products that we're gonna make, of the type of aesthetics that they're gonna have, of the type of behavior they're gonna produce in people. And this is in Spanish because the presentation is in Spanish, but it's from Dynamics in Action, which is a really good book. It's not related, but I think this is really nice, because she said some constraint must therefore not only reduce the number of alternatives, they must simultaneously create new possibilities. And I think we really look into working within the constraints we have, especially in Latin America. We're always like, okay, we don't have the technology, we're not being financed. All this work that is being done in the project is everyone is doing it pro bono or ad honorem. I don't know how it's in English. And this was the finished work that we did. And we actually did this with the people in a workshop that we held. It was like 30 people, and we were all working on the same shapes. And we also worked in texture to see how the color would look with different textures. I actually have all the samples here. I can show you guys if you want. So you have the Pycnoporus, the Ganoderma lucidum with hydrosoluble colorant, with liposoluble colorant, and the white one just to have tested. And this is from the Burley Design Office. It's in Spanish, but I'll translate it. It was originally in English. But I think he has a very nice way of saying, that was in 2008, just how design was going from these manufacturing methods to this biology-based ethics, and how the focus was changing from things and objects to system and behaviors. The values, instead of looking for simplicity, is embracing complexity. And the role of the designer, instead of being an expert designing, it was a collaborative facilitating, where construction is not direct but mediated, where the conditions to stop are almost, instead of being almost perfect, are just good enough for now, which I think it's amazing. It's something that nature does. It just makes it good enough, and it keeps iterating and evolving to make it better for the conditions that it are. And the results, instead of being more deterministic, is less predictable. So now we don't control the outcome, but we just learn about the process and conduct with the process. And the final state, instead of being completed, is just continually being adapted. Sorry, I took longer. And I love David Bohm. If you guys know him, he was a physicist, and he has this great book on creativity. And he's saying that a theory is mainly a way of perception, a way of looking at the world, and not a way of knowing how the world actually is. And I think this is really nice because we keep changing, our theories keep changing, and we change through them as well. Yeah.
Thank you so much, Heidi. That was beautiful. Like, so good to hear like more about your work. We've been like hanging out for such a long time. And yeah, amazing. Like interesting also like conversation were in the chat. Do you have any guys questions to Heidi?
I think we have time for one question because the plenary has started and we do have to bring this to an end, sadly.
Okay. I'm sorry.
No worries. This is great. This is why I haven't said anything in the last 15 minutes.
I'm gonna download the chat because I will look at it later.
Yeah. I also get a copy once this is over,
great.
so I can also share that copy. And I put a lot of it in the notes, but if people remember something, please, there's a section for you to write stuff down. Go ahead. I'll share the notes again.
Yeah, so we're wrapping up then. If nobody wants, like we are out of time basically. Roland had some interesting insights about that he shared.
About what?
Yeah, like picking your minds about like possibilities of DNA storage. That was interesting, and also like you wanted to share work with Suchta. I don't know if I pronounce your name.
Suyata.
Yeah.
No, I was mentioning if we still have time for other people to join, I want to mention Suyata's work because she's here. But maybe, shall I show it? And then Suyata, do you want to tell a bit, Suyata? Like a minute?
Oh, sure. I am completely unprepared, but we can try.
No, but that's fine. That's kind of the idea. It's not about, how do I do this? Share my screen. Share screen 1. Okay, this is a kind of chaos now. Because now you see Hookmine now, right?
Mm-hmm.
Do you guys see something?
Yes.
Yeah, we see
Hookmine.
Or archives, yeah, Hookmine BB.
Yeah, okay. That's then, okay, then I know where I am. I'll make it full screen for you. Microbes rule. I guess I can say that we all know the password. No, I said Suyata's also here, so she might join. To tell something about her work, I hope I'm going to do a project with Suyata, but it's kind of related to this project, but not really related to Suyata, but it's a previous project. And it is at the bottom, I think, when we walk through this. This one, a cure for concrete. Just a whole different type of transdisciplinary research project. Take it away.
Okay, so actually, I don't know how many of you were there at the residency talk that we had, the co-creation talk last night, but one thing I want to say about this project is that I met my collaborator before the residency started. So like, if you see the background that I've got behind me on Zoom, I was taking pictures for a long time of these effects on walls, these destructive, or as I see them, these co-creative effects of nature with man-made. So this is just basically a wall, and you've got fungi and yeast and things growing on it outside. And I had approached this professor. He's a professor of polymer materials, but I knew him through various people. And I had approached him about, so what's happening on these walls? And gradually, it just turned into a project. So he was investigating, so basically what his job is to find uses for these weird waste products that come out of industrial processes. And this particular process that we're focused on here is wastewater treatment. And it's quite tied to the University of Technology in Delft, because they have come up with this wastewater treatment process called Nereda, which you can look up, but it involves bacteria. And so there's an anaerobic and an aerobic process, and there are different waste products from each part of the process. And I've used those to try and decorate concrete, because another of the products of Stephen, who's the professor I'm talking about, is a curing compound for concrete. So what it is based on alginate. And he basically devised in his head that if you put this on concrete, it should protect it while the concrete is setting by stopping the water escaping. And so we combine these two very different perspectives. One's visual decoration, I guess, ornamentation, organic patterns in your environment, which I think is a very important thing. I personally have a problem with bleak concrete buildings. And I would much rather that we have more organic, well-being promoting environment. So these colors are produced by one of the byproducts, which is Vivianite, which turns out to be also a pigment that the oil painters used to use in the past. So it's quite fascinating that this just happens to be one of the waste products from wastewater treatment. It's iron phosphate, hydrated iron phosphate, and its behavior on wet concrete is what you see here, quite an unpredictable, chaotic color change. It's quite difficult to repeat. In terms of the science, it's very hard to know exactly what's going on. But I think as an artist, also having the freedom to play around was a really nice way of us working together.
Yeah, what I like, so what I like about Suyata's project is that what you just described fits into this conversation we had yesterday, indeed, about art science residencies. And then Jennifer Willett, who gave the performance yesterday, said, sometimes when you work as an artist with a scientist, it's also a matter of first having a click with the scientist and then just seeing what they're interested in and kind of look how to incorporate that into your art, right? So also be pragmatic sometimes and because in this case, you found this professor in Delft who did this, and you had this other processes and other ways you were looking at it. So you kind of meet each other in the middle, which is a beautiful way of
exactly. And I didn't know the guy, so as I was saying, is that I still had to invest in the relationship. And we are people; we have to get to know each other as well. And it's not just about being an artist or a scientist or whatever you feel you are. With Stephen, I was lucky because he also loves photography. So he totally got my perspective as well. And I also have a degree in physics, which is how he started as well. So that, it just worked. And here, this picture is of a live installation. This is how we presented it at the end, so that, people can see exactly what you're doing. You're dripping. It's not very sophisticated. You're just dripping a chemical onto wet concrete and letting it set and seeing what the effects are at the same time. We projected this live last year at Today's Art. Anything else? Any questions? Sorry, I wasn't very prepared for that.
Thank you for your thoughtful words, and this looks really beautiful.
Yeah, maybe can I have one? I know we're out of time, but which microbes did you use? So these are kind of microbes used in concrete
I was just, no, so this is the wastewater treatment process is actually patented by a group of people at TU Delft. So I was just trying to look that up because I thought that might, that would be an interesting question, but I don't know. I would have to ask to find out.
A secret patent academic, not open source.
Yeah, I know. Yeah, there's a lot of that at that university. A lot of startups coming from their technological discoveries. Anyway, and Roland, that link you just shared is amazing. Let's work on that. Because I also worked on iridescence on this project, but it was a big failure.
Yeah, I didn't want to share the iridescence, but I want to share this man who has this company in iridescent bacteria, which I shared in the chat. Also had this other, because I've been there at this startup, and he had this startup before where he makes his metal plates with little holes, and then in the little holes you put different bacteria, and then you can it can be used for microbial ecology research, where you see how these colonies of different species react together. And this kind of related to a comment Kenza made to Heidi, that you can also have petri dishes split into 3 or 4 parts and kind of have different species. That I wanted to say. That's really cool, a cool method. I'm just ideating now. I'm really happy to share it. Okay, I'm gonna stop screen sharing. I think you want to take it over again, Carolina.
Yeah, I'm just like, we're like over time, but Barrett told me that like we don't have to stress so much. But like I maybe it's time to go. It would be like I would be personally interested in comments of all of you on like the project skin database, and especially like from like a scientific perspective, how you find it in terms of feasibility and doability. But I also like, we can also like end here and move this conversation to the Slack, because it's like hour and a half and stuff is happening in other rooms. So I don't, yeah, so I don't know what to do. Like if basically we don't have a timekeeper and things that flow. So Barry is going to
Here's
jump
what
in.
I will offer.
Okay.
Since look, there is no time. I have to take over this channel back at 2:15. Here's what I'll do. Y'all can return to the plenary if you would like. I understand. But if you want to continue the conversation here, I'm more than happy to leave this open. And I have to go do other things at this time. But if you want to continue here talking, I will leave it open. I will turn a blind eye and say, like, I don't know. I think there was a great conversation here. Oh, whoops, I accidentally left and didn't close it. That's up to you. So I invite you all to continue to talk. I think this is great. I think we got a great set of people. So yeah.
Oh, we have one more topic left.
Yeah, maybe we can just stop recording it, like, and do it off the record, and then it will be just like more like a hanging out space.
Exactly.
Okay, so I'm hitting stop recording now. And before I do that, I just wanted to thank Kenza, Eloise, Stephanie, Heidi, and Carla for coming here, and Roland and Sujata. So happy, and yeah, I'm hitting stop recording, and now we're going to have a more informal time.